Hey, everybody. Okay, so today we're going to continue our discussion on food webs that we started last class period. During that time, we talked about isotopes and fractionation. We talked about, for instance, carbon isotopes and how those vary among different producers and how that can help us discern relationships between producers and consumers. So we talked a little bit about carbon-13 and nitrogen-15 as being two primary tools that we use when we're talking about isotopes. So for instance, we looked at an example where an herbivore eats one plant. Very easy to discern that relationship because the carbon-13 signature of the herbivore is going to be nearly identical to that of the plant that it's consuming. However, if we have an herbivore that is consuming multiple plants, multiple species, then its carbon-13 signature is going to be some average of the species carbon-13 footprints proportional to the amount that those species make up that herbivore's diet. Okay, so it may not be 50-50 depending if he eats more of the blue one than the yellow, for instance, right? Okay, so how do you know which one is correct? Well, we can utilize these two carbon and nitrogen isotopes together, and that helps us quite a bit. Now, so this is a example looking at a seagrass bed. So we talked about Thalassia before. You'll find it down in West Bay, Christmas Bay, that area. And so this is a fairly simple community. We're looking at three producers, a couple of consumers here, and our grazers in this example. So these are some shrimp and some snails. We can see pretty clearly when we do the analysis that the carbon-13 signature really of these consumers really matches up with the macroalgae that's present in this community, but not so much with the epiphytes and the thalassia itself. Okay, so you can see that if you look at the overlap. So here's our carbon-13 axis down here, and you can see that these pretty much align with each other really, really actually very closely. Okay, so between -20 and -15, remember this is a ratio. This is not the actual amount of carbon-13 present. This is a ratio of carbon-13 to carbon-12 relative to a standard. Okay, so all right, so this lines up pretty well. And when you look at nitrogen-15 here, you can see that it is slightly elevated or enriched above that of the organisms that it is consuming. Okay, but there's really not much of a relationship to these other plants because they're really not consuming them. Okay, so there's no enrichment. They're practically the same level of nitrogen-15, and there's no overlap when we talk about carbon-13. That's not to say these plants aren't important in the overall biologic community. They have their own ecologic niches. They are playing important supporting roles even to these grazers. There's just not a consumptive relationship here. Okay. Now, as we talked about, real life is often more complex when you're looking at these populations. I love the lower level textbooks, especially when they show students data. A lot of times the data that they show is it's so pretty, right? They've got these beautiful S curves or these great Gaussian bell curves, and um, and everything's smoothed out, and it looks really easy to discern. But when we look at real data, that real data can be fairly noisy. The more of it you have, the noisier it can get, right? And the more different parameters that you're tracking that you're trying to put together, that can be even more complex. So this is from a 1997 study, and let's walk through it and see if we can unpack it a little bit. So we're going to start down here, right? And this is in the mid-marsh. And so we've got some Salicornia here. You can discern the producers and the consumers, by the way, by whether the symbol is blocked in like this one is in black. So that makes that a producer, or if it's open, which would make this a consumer. So we've got our two producers here, we've got our consumers. This is a type of wolf spider, by the way, this PAR. Um, and you can see when you look at these consumers, their carbon 13 signature is immediately between the two producers in this graphic, right? So these are largely, they're consuming either, well, their ultimate producers are on either side of those. So their signature represents an average of these two things. Okay. Okay. So that's pretty straightforward, right? The consumer signatures are intermediate between the Salicornia and the microalgae. Okay. If, however, we look at the marsh boundary, right, in this instance, um, this suggests that there might be other carbon sources. So when you look at this, so you got Distichylus up here, right? We've talked about that before. Here's your Salicornia here. I forget what this one is. I'm off the top of my head, but it's in the notes. You know, you can actually find the keys for these. So here again, we've got our old spider. We've got so these are our consumers here in green, circled in green. And you can see that there just really isn't much of a correspondence, certainly not with um, the nitrogen 15 doesn't show that correspondence there. And the relationship isn't very clear even with carbon. I mean, they're in between the Sticulus and then these other two species, but it's not a really clear relationship. And the nitrogen 15 just doesn't make any sense because their nitrogen 15 levels are actually not elevated. They're not enriched. They're actually lower than everything else they're consuming there, right? So this would suggest that in this case, there's got to be some other carbon sources that aren't represented here. That could be detritus or other microorganisms, or particularly in the case of the spiders, they may be traveling elsewhere, like maybe over here, to eat. Okay, so that's a distinct possibility, and it's not, again, that relationship here. You got to unpack it a little bit further before you can see what, and compare it against this one that is certainly more straightforward. Okay, so here, if we look at a tidal flat, then we've got even more going on. Okay, so we've got these little snails here that are these guys here, and these little bivalves right here. And it's pretty clear from the carbon footprint, footprint, I keep saying footprint, I mean carbon signature. I've got climate change on the mind. So, so you've got this carbon signature here, right? And it very much matches up with this microalgae, right? So it's fairly clear that these guys are consuming that microalgae, and in both with the carbon 13 signature, but also with the nitrogen 15 signature, right? You can see the enrichment value there. What isn't clear is if what's happening with these other diets, right? These are a lot noisier, and I would not be able to unpack these and be able to tell you what's happening here easily. So, this could be omnivory, that could be there's planktonic detritus that they're feeding on that isn't represented here, right? This is much more difficult, but this is the reality of the kind of things you're likely to see with real data. So, um, these are not a silver bullet. Okay, so when we're looking at these heavy isotopes, they're not magical in terms of just discerning everything about these trophic levels, but they are a tool. They're a useful tool. They can tell us a lot, but in some cases, there's more going on than those two isotopes are going to be able to inform us wholly. Okay, all right. So um, there are things like omnivory and import and export that can mask food sources. But another thing, thinking back on that last graphic, is that primary producer isotopic signatures aren't constant. So those can change depending on the nutrients that they're exposed to stress levels, to what stage of decomposition they're in. So if they're going through senescence and what stage they're, all this stuff can have an impact on those signatures, right? And then if there's other sources of food there, those sometimes are overlooked, right? So that may not be present in a graphic like this. It might not have been something that the researchers picked up on or tracked, but might have a significant influence here. Okay. All right. So if we were to try and determine, given this scenario, herbivore A has a carbon 13 signature that's similar to plant B, therefore herbivore A eats plant B. Well, is this true or false, or is there not enough information to be sure? Well, we really don't have enough information to be sure because we don't have the nitrogen 15 data to go with it. And so if that's true, then we should see some enrichment there. There could be something else going on in here, and we really, you couldn't tell. All right, different scenario here. Predator A has a carbon 13 signature that's similar to grass B and a nitrogen signature that's slightly enriched relative to grass B. Therefore, Predator A eats grass B. Okay, so is this true, false, or not enough information? It's probably false because it's unlikely that a predator is going to be eating this grass directly. Now, it might be somewhere in that, within that trophic uh, relationship, but that we don't have enough information here to be certain. So I would suggest this is probably false. All right, which of the following are true statements here? You are what you eat. Yes, when we're talking about carbon and nitrogen, that's true. N members contribute more to a consumer's isotropic signature if they are a large portion of the consumer's diet. That is true, right? So if they're eating multiple things, the signatures are going to be proportional to the consumption. Right? C4 plants are usually more nutritive than C3 plants. Well, if you remember back to the last class period, we talked about C4 plants, and the more carbon there is, typically, number one, the less palatable they are, the less tasty and delicious they are, and they tend to have a lower nutritive content as well. That's why in restoration projects, a lot of times, especially in freshwater environments, we'll use plants that have they tend to fall in these C4 categories because they tend to survive longer because nobody's eating them in order to establish the marsh, and then other plants will volunteer in those communities and fill it out over time. Okay, so back to our question here then. So that one would, so not C, right? A and B, we've got are good ones. In a food web ecology, the nitrogen signature of a consumer is typically higher than the nitrogen signature of its food source. This is true, okay? And then overfishing typically alters community structure through bottom-up pathways. Fishing is a top-down pressure, okay? Okay, so with these complex interactions, again, these marsh communities can be, plant communities can be quite complex, and a lot of different relationships happening there. So it isn't always really easy to discern what's going to happen. And so when we start looking at these interactions and asking questions, we see that things, again, don't always work out the way you might, they might seem like they would on the surface. So, for instance, are more resources always better? What happens if resource supply, which is a bottom-up effect, right? What happens if that's altered? Well, nutrient enrichment should increase primary producer biomass, right? Because that's, you're increasing the food, so that should be good for the things that eat the food. Okay, so they're herbivores. However, nutrients can also shift the composition of the producers that are present in that community, right? Either by, well, most of the time, it's going to be by how many of each different species there is. One might benefit more from that nutrient addition than others. Okay, is that necessarily going to have a positive effect on those herbivores? Well, so this comes from a study that was done out on the West Coast. It's an older study, but and they looked at the effect of nutrient enrichment on a benthic microalgal mudflat community. And so under normal conditions, you would have a community that the mudflats are typically dominated by diatoms, And so you get a kind of a picture of that here in this picture to the left. Whereas in enriched conditions, that can stimulate the growth of cyanobacteria, which can form these really thick mats on the surface of the mud, like you see over here in this picture to the right. So they looked at running an experiment utilizing these natural consumers in these mudflats, these snails, and and what they wanted to do was evaluate the effects of this nutrient addition. So their experiment was really pretty straightforward. They just set up plots, and in some of the plots they had added nutrients and they had added snails, and some they only added nutrients. In other instances, they didn't add nutrients, but they added snails. And in some instances, these were ambient conditions minus the snails altogether, right? So what did they find here? Well, what they found was really kind of an interesting thing because the snails typically are consuming that benthic microalgae. And so while it, again, seems like that would be a good thing, what they found was that the mortality of those snails was actually higher where you had the nutrients than in the ambient conditions, which is kind of counterintuitive because you would expect more food means more snails, right? More food means more herbivores. So, this was kind of they started thinking about it and looking at it. What they determined was that by fertilizing these plots, it created this increased biomass, but that biomass included cyanobacteria. And this actually covered the diatoms, which are the more common food source for these particular snails. Okay. So instead of eating the diatoms, now the snails started eating the cyanobacteria. And the cyanobacteria, number one, is some mildly toxic. Um, but the other thing is that that mat that forms when that cyanobacteria creates that mat that I showed you in that prior picture, that creates anoxic conditions below the mat, and that in turn can negatively affect the snails as well. So we can see the relationship here between the snails and the cyanobacteria by looking at this pigment that's found in that cyanobacteria. And so we see that it increases where they added nutrients compared to where there were no nutrients. And again, this had this negative consequence that is a little, again, counterintuitive. Okay. All right. So, what about interactions where you might have top down and bottom up forces that are working either in conjunction with or against one another? So, an example of this would occur, for instance, in mangrove communities where you have bird rookeries. This very blurry picture here actually is this is from an area over by Winnie, East Galveston Bay. And I'm sorry you can't see this very clearly. I quite obviously blew this picture up a little bit too much. But in these trees here, you can see bunches and bunches of birds and nests, and that's typical of these rookeries. They utilize the trees to get up off the ground. So this would include things like spoonbills and ibis and a variety of different egrets and herons. And they all are intermingled with each other. So their nests are all right next to each other. There can be tons of birds, and it is a very nutrient rich environment, if you will. Now, this study didn't occur in that particular marsh. This is an older study done in the 70s. But it illustrates, I wanted to illustrate what these rookeries look like. But so these mangroves in this particular study, they looked at a couple of different metrics here. And so this graph's got a lot going on in it. But if you look at it, they're tracking the flower production, branch production, and leaf production in these mangrove species, both in mangrove areas that had rookeries present and some that didn't have rookeries present, right? So generally where you have rookeries, they're very concentrated, and where there aren't rookeries, you may have a nest here or there, but you're really not going to have very much. And that's very different in terms of the nutrient enrichment. Because where there's a lot of birds, there's a lot of poop, and also there's a lot of fish material because these are mostly fish-eating birds. Um, well, partly fish-eating birds. I should say they're all fish-eating birds, but mostly fish-eating birds. So especially the herons, egrets, and those kind of things, and they'll bring those back and regurgitate them for their chicks, and so that gets spread around, and that has a lot of nutritive value in it. And so they were tracking this, and they tracked it over time, both when the birds were present and when the birds were absent. And it was kind of an interesting relationship that they uncovered because what they found was that the nutrients helped increase the amount of production for the flowers and the branches, but it wasn't as clear with the leaves. And it was so it came down to another factor, which is herbivory, right? So here we've got top-down pressure. And this herbivory damage was much higher on the enriched leaves than it was on the non-enriched leaves in the sites that didn't have these rookeries. And that's because those enriched conditions, all of that poop and fish meal that was fertilizing those mangroves made the leaves very nutritious, and that attracted the attention of insect herbivores, primarily Lepidopteran larvae, but also some other insects as well. And so that herbivory was really high when the birds were there, right? They were producing a lot of nutrients in those areas, and that really elevated the tastiness and nutritional value of the leaves, which then increased the number of herbivores. And that kind of went away after the birds moved on at the end of the breeding season. So with the leaves, that herbivory pressure actually outweighed the positive growth effects of the nutrients. And so, but not so much with the fruit or the branch production. This was really only the leaves because that's all the insects were eating, right? Okay. So, what about non-consumptive interactions where the plants maybe are just serving a form of refuge, right? Food webs aren't always structured on consumptive interactions or maybe not completely on those consumptive interactions. So, uh, here we're going to look at a seagrass system and the effect that seagrass has on these fish. And the interesting part of this is that like I said, not all of these interactions require these consumptive relationships. Sometimes the presence of a species can alter other interactions within that community, as we'll see here. So this one looked at predator and prey. So we've got these killifish, and so these, I think these are Gulf killifish. Which I'm struggling to remember the scientific name over off the top of my head. But anyway, and then also some sea trout are your predators in these environments, or one of the primary predators. So in environments, they set up this experiment and they just looked at, well, where you've got uh, just, where you don't have any predators. So we'll start over on this side, right? Where you don't have any predators, their oxygen, their respiration rate is low, right? They're not stressed. They're not having to consume a lot of oxygen in order to move around and evade predators. There again less stress there, and that reflected in their ability to put more energy into biomass production. Right, so they grew faster. Oh Lordy, I must be getting tired. Have some coffee here. So they're growing at a faster rate where these predators are absent. Okay. And there can be other aspects to that as well. So part of it might be energy production. Part of it might be that the bigger ones are easier for the trout to see and they're consumed faster. Right. Okay. So where you had the predators, okay, and no seagrass, then here the oxygen consumption by the killifish was much higher. Okay, you can see the comparison here on either end of this, and the growth rate was much lower. But when you add seagrass to this equation, where they had seagrass present, this provides refuge for the killifish, and as a consequence, their respiration rate, rather, oxygen consumption went down considerably. Actually, looked very similar to what it was like when there were no predators present. And the growth rate also was much more similar to that where in the control, right, where there were no predators present. And here, remember, we're looking at these error bars here, and you'll see that there's an overlap between the top error bar here and the bottom error bar here. So statistically, this likely would not be a statistically significant difference here between those two, but there would be a difference here. Okay, so same thing up here, right? So this plant, the seagrass, neither of these species eat the seagrass. There's no consumption occurring here at all. They're not consuming the submerged aquatic vegetation, the SAV, right? Rather, or SAV, in this instance, that SAV is simply providing structure, and it's altering the relationship between the predator and the prey in a way that you can measure here. So again, there's a lot going on in these food webs, a lot of complex, both direct and indirect interactions. And ultimately, when you're looking at these food webs, you have to take all of these things into mind to be able to really fully understand that. So, with that, when you get to your test, these are things that I would expect you to be able to understand and recount as you go through there. So if you're not able to do that, please reach out to me, and I'm here. I'm happy to help you if I can, and we look forward to talking to you. So with that, I will see you next time.